So I'm going to talk about crop wild relatives, and I'm going to talk about food security. I'm sure you've heard about food security already this week, the policy context, why we need to look at crop wild relatives, and I was asked to focus specifically on UK crop wild relative conservation. So I'm going to talk about that development of conservation plan for UK crop wild relatives action plan, and possibly the implementation of the first genetic reserve in the UK, which is going to be on the lizard. And I should have also mentioned my co-authors, Hannah Fielding, who's a researcher in my group, and Julian Hoskins, who works for Natural England. So why conserve actively crop wild relatives now? Now, I don't know if anyone else has showed this slide before or made the same point, but the major problem we have is too many human beings. Today we have 7.31 billion human beings in existence and predicted to be 9.6 by 2050. These are the UN figures, though there are various scenarios. But the basic figure that the UN comes up with is 9.6. To feed this increased number of human beings by 2050, requires globally an increase in food production by 60%, and that is 100% in developing countries. In the same time scale, we have a predicted reduction as a result of climate change of 2% per decade over the same time period. So we have increasing mouths to feed, we'll be able to produce less food. So in other words, the scenario is that we should be improving agriculture. If we don't, then we're going to end up, which is shown in this slide, with the harvest shortfall, which is human misery, malnourishment, starvation, civil strife. So we need to be very active in meeting this demand, otherwise we are going to have serious problems in the future. Now, one way of tackling this, and obviously I stress it's not the only way of tackling food security, is systematic conservation and use of crop wild relatives in breeding. Why are crop wild relatives particularly interesting? Because they have a wide genetic base. They haven't gone through the domestication bottleneck. They're tried and proven, lots of examples of pest and disease resistance from using crop wild relatives in breeding programs. I'll show you some examples in a minute. And with technological advances, it's becoming easier and easier to use crop wild relative diversity. The importance of crop wild relatives is also increasingly recognized. Just a couple of major policy documents, the strategic plan agreed in Nagoya in 2010, Target 13 talks about by 2020, the status of crops, livestock genetic diversity in agricultural systems, and their wild relatives. So wild relatives, interestingly, picked out for specific mention of their importance in terms of meeting the targets of the CBD. In the Global Strategy for Plant Conservation, again, wild relatives specifically picked out as important as a target for conservation. And in a sense, they're the perfect marriage of conserving wild species and their use in promoting the Millennium Development Goals, as in linking poverty alleviation and the reduction of extreme hunger to conservation of the biosphere. So what are crop wild relatives? So crop wild relatives are the wild species that happen to be relatively closely related. Some are closely related, some are less closely related. They form the ancestors of the crops. They're now of interest to us as gene donors because we can take traits from these wild species and transfer them into the crops themselves. And there's an awful lot of novel genetic diversity contained in these crop wild relatives. How do we find them? In the broadest sense, we define a crop wild relative as any species, any wild species found in the same genus as a crop. That comes up with a very large number of species. For instance, if you look at the flora of Europe, if you use that definition, any wild species found in the same genus as a cultivated species, then you end up with 80% of the European flora being crop wild relatives. So if we want to specify that more precisely, we can focus on those used in food and agriculture And we can use those that are most closely related, as in identified by trying to cross the wild species with the crop. So we use gene pool concepts, and we come up with this much narrower definition. Then we start getting down to more manageable numbers of species that can be used in breeding. So examples of sources of traits. So this is just taking wheat. These are wild relatives of wheat. You can see a mass of wild relatives here on the left-hand side, and the various traits that have been used, derived and crossed into wheat to help us increase yield, etc. And also I've shown here, this is the crop wild relative Aegilops spartoides, a small grass, insignificant grass found on Greek islands, Turkey, and the benefit. That presents the bee genome that has been used to create the hexaploid wheat. So huge benefit from use of crop wild relatives in the past. The bottom line, and this is what the politicians like to hear, is how much are these things actually worth? Give me a dollar value. So the dollar value, this is an old figure estimated in 1997, for global use of crop wild relatives annually was $115 billion. $115 billion. We're talking about a lot of money involved in utilizing the genetic diversity found in these wild species. Now you would assume with a resource that's so fundamentally useful, has such high value, that it wouldn't be threatened and we would already be doing systematic conservation. So if you look at this, first of all, if we take Europe as an example, we did a study funded by IUCN looking at the top 572 crop wild relatives in Europe. We did red listing, as I think the previous speaker was saying about doing red listing, and we found that 16% of crop wild relatives, these priority crop wild relatives, are imminently threatened with extinction. Now, interestingly, this is the thing I wouldn't tell a non-specialist audience, the general figure that's quoted for plant species is 20% of all plant species are threatened. So why do you think crop wild relatives are less threatened than the average plant species? My guess is because many of them are found in disturbed habitats. And so they have more strategies for coping with the stress of modern life. It's a guess, I don't know, but that's the reality. Okay, so if we look then at ex situ conservation, and these figures again are European figures rather than UK figures. So about 10% of gene bank accessions held ex situ in Europe are wild species. A fairly high percentage of those would be crop wild relatives, but they only represent 6% of all the crop wild relatives found in Europe. So 94% of crop wild relatives in Europe have no accessions conserved ex situ at a European scale. If we then look at in situ, well, you can say, look, there are protected areas everywhere, and there are crop wild relatives found in those protected areas. But the problem with that is they're passively conserved. Unless you are actively conserving that specific species, you're managing the site overall. And so you could make management changes that will change and favor one species over another species. And because you're not monitoring the crop wild relative could be lost in time. So if we then look at active conservation of specifically focused on crop wild relatives, there are basically a handful, less than 5 globally, sites in the world where this is being done. And I've listed them here, the main ones here. The only one in Europe is this one in Madeira, where they're conserving a wild relative of beet. And in that case, it's a perfect example. It's a species that's endemic to an uninhabited island off of the coast of Madeira, and nothing is going to change the population anyway, because there's no habitation, there's no animal, no goats, sheep, etc. on the island. In terms of the causes, you probably can't read these, but the causes of threat to crop wild relatives are industrial agriculture, tourism, what else we've got here? Housing and urban development. So these are the various threats that are threatening crop wild relatives at the moment in Europe. Okay, so how do we set about conservation of crop wild relatives? So there are numerous different approaches to conservation. We can do ex situ, in situ. You should do both as a complementary approach. But you can think of it like a series of Russian dolls. So you have one, if you're doing in situ conservation, you have one protected area. And then that protected area fits in the next level, which is the national network of protected areas, conserving crop wild relatives. That fits in the regional network of protected areas that then fits within the global area and associated with that. So that's for in situ, and you can also have the complementary approach for ex situ as well. So these are a couple of those global sites where they're doing in situ conservation. This one is in Syria. God knows what's happening in this site at the moment in northern Syria. But this is an analysis we're just about to publish where we've looked, trying to identify the top 100 sites around the globe where if you had money to put into setting up a new protected area or take an existing protected area and change the management to make it focus on crop wild relatives, then where would you site? So the green triangles are the locations of these existing protected areas where we want to promote conservation, and the hatched areas are the so-called Vavilov centers. The Vavilov was a scientist who died in the late 1930s, and he identified 8 centers of crop diversity around the globe, and those are primarily the areas that these centers are located in. Right, okay, so let's move on now to looking at national level. So as I said, one of the levels is national level. And we came up with this basic schema for how to do national level conservation of crop wild relatives. So you have to start off by identifying a checklist, a complete list of all of the crop wild relatives that are present. Then you prioritize it. If you come up with a list, say, of several thousand, you can't conserve several thousand species all at once, so you want to focus on the most important ones, so you prioritize them in some way. You come up with your inventory. The inventory are the ones that you're going to collect your detailed data on. So these are ones you collect your detailed data on, and then you implement that doing either in situ or ex situ activities. And the bottom line here is use. The difference between crop wild relative or agrobiodiversity conservation and other kinds of conservation is it's explicitly linked. Conservation is linked to utilization. We're conserving for the benefit of humankind, for exploitation, if you like. Okay, so let's look specifically at the UK. So we started off with the UK checklist of crop wild relatives. So how we got that is we had an existing project 5 or 6 years ago funded by the EU, and we developed a European crop wild relative checklist. And then we basically just filtered that for all the species that were found in the UK and came up with this list of 2,109 crop wild relative taxa species, subspecies found in the UK. We then prioritized them, and the criteria we used for prioritizing were whether the crop, how useful the crop was, native status. We wanted to conserve crop wild relatives that were native. We wanted to look at the actual value of the crop. So the higher value the crop, the more crop wild relatives within its gene pool we'd look at. How close the crop wild relative was to the crop, whether you could cross it easily or was it more difficult if it was more remote, whether it was threatened, and the conservation designation. So obviously within the UK we have some species that we know already have conservation priority because the government they're in, Schedule 8 is it, of the Wildlife and Countryside Act. Now the important other step we did is we didn't just decide this as a research group in Birmingham, we involved various stakeholders. So let me tell you a story. Originally we did this research, we did the UK crop wild relative analysis in 2007. I did it with one MSc student. It was a quick piece of work. We published it and said, right, these are the places where you need to conserve crop wild relatives in the UK. I then very arrogantly went along to Natural England and said, aren't you so pleased that I come up with this list of where you should conserve crop wild relatives? Let's get on with it and do it. They said, first of all, we're not interested in crops. And secondly, you have to do the work jointly with us, otherwise we won't do it. So we've gone through the whole process again, which has taken us another, another 9 years to complete this work. But basically we've come up with our inventory, which is 223, which you can see here. This is a list of the number of genera, number of species, intraspecific taxa, and whether their native status, whether they're native, neophytes, archaeophytes, etc. Then we did gap analysis. So gap analysis is a comparison, basically, once you work out which taxa you're going to work on, a comparison of diversity that exists in nature against the diversity, the sample, which is conserved using existing in situ, ex situ techniques, and then helps you identify where are your gaps, and your gaps then become priority for future conservation action. So then, this is Hannah Filder, who did a lot of the work, and she's identified the top, I think it's 27 sites in the UK for conserving crop wild relatives, and the best site is down here on the Isle of Purbeck, which has 124 of the 223 priority crop wild relatives present. The second site is in Cambridgeshire, has 17 additional species to the 124, so those 17 are not found in the first site. It also has 104 in common with the first site. And the third site over here is in Wales, which has a further 11. So that's our priority for doing in situ conservation. So obviously the Isle of Purbeck is our highest priority. And then the second site, second, etc. In terms of ex situ analysis, we looked at the 223 priority crop wild relatives. We found in the UK we're conserving just over 1,000 accessions of 146 of those 223, and the 3 gene banks that are conserving them are the Millennium Seed Bank, University of Warwick, and University of Aberystwyth. The ones that had the largest number of samples, perhaps not surprisingly, are the ones that are also forage species, commercial forage species, so perennial ryegrass and white clover. We have large numbers of collections, but we did find 77 taxa with no stored accessions. So 53 of these are introduced taxa. Now, when Hannah first got this result, I said, no, you must be wrong. We must have all this material in the gene banks because, sorry to criticize Kew, but Kew have said that we're conserving the UK flora. So when we actually looked at this, when Kew are collecting material from the wild and putting it in the gene bank, what they're focusing on is only native species. So they're excluding archaeophytes and neophytes. So an archaeophyte is a species that arrived in the UK before 1,000 years ago, and a neophyte is one that arrived between 1,000 and 500 years ago. Now, in that time that they've arrived, if it's 500 years ago, obviously they've changed genetically, and so there's a lot of genetic diversity. So we would argue strongly, and I'm glad to say that Kew have agreed with this, they're going to change their policy and start collecting archaeophytes and neophytes as well. But basically, the 3 species of that 223 that are already well conserved are white clover, perennial ryegrass, and Plymouth pear, which is a very restricted species anyway. Okay, so why the lizard? We worked on the lizard because it's a warm climate, complex geology, range of soil types, a lot of variation in microclimates, varied use pattern, and a lot of the area is already well conserved. And in terms of the UK, it's the 6th best site. Now, as well as looking at the UK, we also did each devolved administration separately. And if you do the gap analysis prioritization for England alone, then the 2nd highest site is the lizard. And so these are some of the wild relatives that are found on the lizard. These are the sites where we're sampling genetic diversity and looking at the patterns, comparing genetic diversity on the lizard to outside of the lizard. And this is a report we published saying how important it was to do crop wild relative conservation in the UK. So similarly, we're doing this similar work in various other countries in Europe and also outside of Europe. And interestingly, it's much easier, it seems to be much easier to get funding to work outside of Europe than it is inside Europe. Yes, okay, I'm finishing. So just to finish up then, just to say we've started to set up the site on the lizard as the first UK crop wild relative genetic reserve being managed by Natural England. The important step we need to take is actually changing the priorities within SSI's special areas of conservation, national nature reserves, so they prioritise crop wild relatives. We're in the process of doing that. And have formal recognition of genetic reserves within the UK. We need to get on with establishing the site at Purbeck and take these other actions or priorities for the future. Now, I'm not going to mention this, but this is just to reinforce the point that you need to make this link between individual reserves, national networks, regional networks, and global networks. And this is some work we're currently doing for FAO, designing a global network on the basis of this diagram here. So our take-home messages, very simple, significant value in crop wild relatives. They have huge potential for utilization. They are threatened. We need to value this plant resource that we have, this botanical diversity. We need to be active in terms of in situ, ex situ conservation, both within the UK, but also at regional, global level, and revisit this link between conservation and use. Because as I stress, the last words, as I stress, the importance of conserving crop wild relatives is not to get them into the MSB, but it's to get them out of the MSB so that people can actually use that material in breeding programs and get benefit to the people who live in the UK. Thank you very much, and sorry for overrunning.
Thank you very much.